Hello everyone, welcome to my channel LifeScience with Nivedita. This is KSET LifeScience 2024 Key Answers video part 2. I already made a part 1 video where we discussed about 50 questions solutions. So in this video we are going to discuss about remaining 50 questions solutions. Okay, so before going to start this part 2 video of this LifeScience solution video, let me clarify about the things which I made in the part 1, that is the previous part. So I made mistakes in two questions, that is the question number 20. So the answer for this question number 20 is option C, that is bulbourethral gland. Okay, so I made a mistake. I did this. Option B is the correct answer. This is what I said in the previous video, but correct answer for this question is option C. Okay, and similar like this, one more question, that is question number 50. Second. Okay, so yes, so here you can see in the question number 50, that is the term evolution was first used by. So I said this is option C is the correct answer, that is Darwin. But here option B, that is Herbert Spencer, is the correct answer for this question number 50 question. Okay, so sorry for this mistake. I made a mistake on these two questions. So a special thanks to my viewers because they let me know in the comment box. Okay, so thank you so much. I really like the discussion which you people made in the comment box. Even no, that thing makes me to motivate to do more video like this. Okay, so now in this video we are going to solve remaining 50 questions. So in that 50 questions, the first question is what, that is 51 question. Let me read the question. The change in allele frequency over a period of time within the population is known as what? Okay, so here the change in allele frequency over a period of time within a population is known as, once again, let me zoom this. Okay, so 51 question. Option A mega evolution, option B micro evolution, option C macro, as well as option D co-evolution. So here answer for this 51 question is, that is option B micro evolution. So here micro evolution means that is a change in process like mutation, selection, gene flow, etc. So this thing that changes in the process is called as micro evolution. Okay, so next question, that is question number 52. The raw material of evolution is what? So here the raw material of the evolution is mutation. So that's why option A mutation is the correct answer. Okay, so next 53 question, that is Mueller was the first to produce induced mutation in. Okay, so Mueller was the first who will produce the induced mutation with the help of, that is Drosophila. Option A, Drosophila is the correct answer. Drosophila is a fruit fly which is used for breeding experiments and cytogenetics, etc. Okay, so here Mueller used Drosophila used to produce induced mutation. Okay, so the next question, that is question number 54. Origin of species was written by. So this is a, we can say this origin of species is a foundation of evolutionary biology. Okay, so this book was written by the scientist Darwin. That is option D, Darwin is the correct answer. Okay, so next question number 55, that is species inhabiting different geographical areas is known as what? Okay, so here answer for this question is, that is option B, allopatric. So here allopatric means the organisms, the species which are separated by a physical barrier. From the physical barrier, the species are separated from one geographical area to another geographical area. Okay, so now move on to the next question, that is question number 56. Okay, question number 56. Which geographical epoch period is marked by dinosaur dominance? So this question based on geological time scale. Okay, so option A, Paleozoic, Mesozoic, Cenozoic, Proterozoic. So here answer for this question means in which a period the dinosaur is dominant. That is option B, Mesozoic era. That is Mesozoic era. This takes place, this Mesozoic era, that is about 253 to 67 million years ago. This is the time period when the Mesozoic era was happened. So here, when reptiles, especially dinosaurs, in the Mesozoic era, especially dinosaurs ruled the world and also called as golden age of reptiles. This Mesozoic era also called as golden age of reptiles. Okay, so answer for 56 question is option B, Mesozoic era. Okay, so next question, that is question number 57, that is which of the following is essential for fermentation process to occur. So in the anaerobic fermentation, especially in the anaerobic fermentation, the essential thing is, that is ethanol. Okay, so that's why option D, ethanol, is the correct answer for question number 57. Okay, so now move on to the next question, that is question number 58. So which of the following vaccines do not have age restriction for administration? So here you can see not, means which vaccines not have age restriction. Anyhow, MMR vaccine, that is measles, mumps, and rubella vaccine. So this vaccine has age restriction, even though this is you know given by two dose, that is first dose and second dose. First dose is given in 12 to 15 months, and second dose, that is 4 to 6 years. Okay, so next is the polio vaccine. Yes, this polio vaccine is also age restriction, that is under 5 years. Next is the HPV vaccine. HPV vaccine means human papilloma virus vaccine. So this is also age restriction for administration, that is 11 to 12 years. So last one, that is rabies vaccine. So this rabies vaccine does not have any age restriction. Okay, so that's why answer for this question is what? That is option D, rabies vaccine. So rabies vaccine have no age restriction for administration. Okay, this question is based on immunization schedule. Okay, so next question, that is question number 59. Biofortification used transgenic animals by gene manipulation can be achieved by carrying out which one of the following? That is, the biofortification process, that is to improve the, any composition, that is nutrients, any organic substance, any quality, whatever, that is what called as biofortification. So here, to improve what, this biofortification is used in the transgenic animals. You can see this is in the transgenic animals. So answer for this question is, that is improving carcass composition. Improving carcass composition. Okay, sorry. So here carcass means the meat. If it is the meat of that is the two, especially the dead meat, which is rich in organic substances, or in to increase the meat quality, a biofortification process will done. That too for transgenic animals, biofortification will done. So answer for this question number 59 is option C. Okay, so now move on to the next question, that is question number 60. Yes. So the use of genomics in diagnostics has significant benefits in terms of quickly identifying disease etiology for. So here, usually that is genomic therapy for chromosome transplantation. That for that thing. So your answer for this question number 60 is option C, X-linked disorders. That is for genomic therapy. Example, some, if any X-linked disorders is there, to find out that we can use this genomics. And one more thing, in trisomies also genomics is used as a diagnostic tool. So in 60 question, I had a doubt in between this option C and option D. Option C or D. Okay, so I'm not sure which one is correct, but In option C and option D, I had a doubt. Uh, Let me clarify once final key answer will release. Okay. So next question, that is question number 61. That is, resins of medicinal value are an example of what? Okay. So here option A, primary, secondary, value-added plant product and biofortified entity. So here answer for this question number 61, that is option B, secondary bioresource. So here secondary bioresource means produced as a byproduct. Okay, during the processing what happened, which is produced as a byproduct. So here this resin is there, right? This resin is extracted from the secretion of the plants and trees, and which is then used as a flavoring agents, diuretic agents, etc. That's why this is coming under secondary bioresource. Okay. So now move on to the next question, that is question number 62. That is, thriving of diverse living species on the earth is solely due to. So here the answer is option A, inbreeding. Inbreeding means that is which is helps in accumulation of superior genes and which will increase the diversity. Here where the increase the diversity takes place, that is in the inbreeding. Okay. So the next question 63. At which stage of waste treatment do methanogenic microbes play a role in detoxification? So here in the sludge digestion, this one, conversion of organic matter into biogas, that is where this is a stage where waste treatment to microbes play a role in the detoxification. So answer for this question number 63 is option C, sludge digestion, that is conversion of organic matter into biogas. Okay. So now move on to the next question, that is question number 64. So what are the elements of biosensors? So here the some order there, we have to place it in the order wise. The first one is what, that is elements of biosensor, that is analyte. So after the analyte, that is after the analysis, that is bioreceptor. So after this bioreceptor, transducer. So at last, after the transducer, it has to measure the signal, that is measurable signal. So here you can see the first one, analyte, receptor or bioreceptor, transducer, signal, output. Okay. So 64, the answer for the question number 64 is option B, that is analyte, receptor, transducer, signal, output, that is measurable signal. Okay. So this is about question number 64. Okay. So next question, that is question number 65. Yes. 65. Okay. So in the image given below, you can see the image here. Different conformations of plasmid were resolved in 1% agarose gel. Identify which conformation migrates slower to faster. Okay, so here which okay, 1% agarose gel. So which conformation migrates slower to faster? So here we have to find out the conformation, which is linear, nicked, supercoiled, like that. Okay, so here you can see this one, the first line is there, right? That is nicked one, which contains the nick conformation, and this one is linear. So the dark one, you can see the dark one here, this is supercoiled. This is supercoiled. Okay, so answer for this question, that is question number 65, that is option D. First one is nicked, then linear, then supercoiled, that is from slower to faster. Okay, so this is about question number 65. So now move on to the next question, that is question number 66. Yes, 66. So what is the main limitation for using Edmond degradation method for protein sequencing? So here to find out the protein sequence, this is one of the method, that is Edmond degradation method. So answer for this question, you can see, which is here, limitation is as, that is, it cannot determine N-terminal sequence. Yes. It requires a large amount of protein. No, it cannot sequence proteins with modified amino acids. No, it cannot be automated. So here, in the 66 question, the question number A is the limitation, because the Edmond degradation proceeds from the N terminal of the protein. It has to start from the N terminal. For example, this is a protein. Okay, the protein has to start from N terminal and ends with the C terminal. It has to proceeds from the N terminal of the protein. So here the Edmond reagent will react. Where in the N terminal, Edmond reagent will react. So because of the reaction takes place in the N terminal, that is with the amino acid, the N-terminal sequence, which is cannot determine. That is one of the limitation in the Edmond degradation. Okay, so this is about question number 66. So the next one, that is question number 67. What does the total sum of squares in ANOVA represent? So here ANOVA is nothing but AN means analysis of, VA means variance. So here the total sum of squares in ANOVA will represent total variation in data. Okay, that is analysis of variance, which indicates the total variation of data. That's why option C is the correct answer for question number 67. Okay, so now question number 68. One second. Okay, if the values of two variables move in opposite direction, the correlation is said to be. If it is, for example, let me draw the graph. If two variables are move in the opposite direction like this is called as negative. This is the positive one, that is positive correlation, and this graph represents no correlation. So in this question, that is correlation, that is in the opposite direction, is said to be the negative correlation. So that's why option B is the correct answer for question number 68, like this graph, that is negative correlation. Okay, so now move on to the next question, that is question number 69. Okay, 69. Radioactive isotopes used in Hershey-Chase experiments were, so radioactive isotopes of these elements were made it easier to trace them during the course of experiment. So in this Hershey and Chase experiments, two radioactive isotopes are used, that is phosphorus 32 and sulfur 35. Option C contains phosphorus 32 and sulfur 35. Anyhow, DNA contains phosphate backbone, that's why phosphorus 32 as a radioactive isotopes, and some amino acids contain sulfur in its side chain, that is cysteine and methionine, that's why sulfur 35 they choose as a radioactive isotopes. Okay, option C is the correct answer for question number 69. Okay, so next question, that is question number 70. So freeze fracture method for microscopic examination does not involve. So here we have to eliminate the wrong one. So here rapid freezing, yes. Fracturing, yes. Cytoprotection, no. Sublimation, yes. So usually, freeze fracture method, this is the process by which chemical compounds provide the protection of cells against the so here this freeze fracture method used to study the structure and function of cell membrane. Okay, this one. So when come to the steps of this freeze fracture method, which includes rapid freezing. So after this rapid freezing, fracturing. After the fracturing, that is vacuum sublimation, that is sublimation of ice. Vacuum sublimation of ice. So after this vacuum sublimation of ice, replication and replica. cleaning. So here you can say rapid freezing is there, fracturing is there, sublimation is there, but cytoprotection, there is no question of cytoprotection, because this cytoprotection is a process by which chemical compounds provide protection of cells against harmful agents. That's what called as cytoprotection, protection of the cells against harmful chemical agents. Okay, so here cytoprotection is eliminate from this freeze fracture method. That's why option C, cytoprotection, is the correct answer for question number 70. Okay, so now move on to the next question, that is question number 72. So the next question is 71. PET scan techniques are used to study what? So PET means that is positron emission tomography, which will produce 3D image of inside the body. So here, this PET scan technique is used to abnormal activity in our body. Anyway, in the in the very less kind, that is in the very rare time, to the progress of pregnancy also PET scan will use, and also the activity of babies also find out by PET scan. Here all options seems to be the correct, but option A tends to more when compared to others, that is abnormal activity in our body, because this PET scan used to find out the brain functions. Anyway, it also used to scan the tumors in the body to find out the cancer tissues and all. So that's why when compared to the remaining options, this A option will seems to high yielding. So that's why maybe option A is correct, but we have to wait till a final answer key will release. Okay, so in my point of view, option A is correct. Anyway, option D is also seems to be correct. Sometimes option B, C also correct, but this A option will yields more. Okay, so this is about 71 question. So now move on to the next question, that is question number 72. Okay, so this question is based on population size. Let me read the question. In a field study to estimate the number of mongoose in a locality, 60 mongoose were initially captured, marked, and released. 80 mongoose were captured after 1 month from the same field, of which 10 were previously marked. From these findings, estimate the population size of mongoose in the field. So here we have to find out the population size. So first write down based on the questions. In the field, estimate the number of mongoose in a locality. 60 mongoose were initially captured. Okay, so here N1 is 60. That is which is caught in the first time. Okay, so N1 is 60. So okay, marked and released. Then 80 mongoose were captured after 1 month from the same field, of which 10 were previously marked. So after 1 month, 80 mongoose were captured. In that 80 mongoose, 10 were previously marked. So now let me write N2, that is total caught on the second time. In the first time, 60 mongoose were caught. In the second time, that is, one second. Okay, so here we have to use the formula, that is, okay, N1 is 60. The next one is, that is, N is equal to MT by R. Okay, so here this N is the population size, and M, that is total marked, and total mark, that is M, is nothing but N1, that is 60. Okay, so M is what? That is total marked one, and T, this T represents the total recaptured on second visit. In the second visit, how many mongoose were recaptured? And this R represents the number of mark recapture, number of marked one. Okay, so here M is what? That is 60, which were initially captured. Okay, so next the T is nothing but what? That is total recapture on second visit. You can see on the second visit 80 were captured, that is 60 into 80. And here this R represents what? This R represents number of mark recapture. That is, you can see 10 were previously marked. That is 10. Okay. So here 60 into 80, that is 4800 divided by 10, 10, 10 cancel. So the N, that is population size, is 480. Here you can see where 480 is here, that is option B. 480 is the correct answer for this question number 72. Okay, so this is about the population size related question. So now move on to the next question, that is question number 73. Okay, question number 73. So which of the following amino acid is a precursor of niacin? So here to produce the niacin, which amino acid is precursor? So here answer for this question is option C, that is tryptophan. Usually, when come to the amino acid metabolism thing, this tryptophan which is present which is in the presence in the form of iron, with iron, this has to convert it into kynurin, then quinolic acid. From the quinolic acid it has to convert it into nicotinic acid. From the nicotinic acid it has to convert it into NAD. So at last from the NAD it has to convert into niacin, which is the precursor of tryptophan amino acid. Okay, so now move on to the next question, that is question number 74. The isotopes of a neutral atom of an element show variation in which of the following properties? So here, once again, okay. So isotopes of a neutral atom of an element show variation in which of the following properties? So usually for neutral atoms of isotopes, the same element, these have the same number of protons and electrons, which is also equals to the elements of atomic number. But here the only difference is what? Which are differ in the number of neutrons which affect the atomic mass, that is mass number. Only variation takes place in the mass number. So that's why answer for this question number 74 is option D, mass number. Okay. So now move to the next question, that is question number 75. So one of the following thermodynamic law gives the concept of enthalpy. So the concept of enthalpy is explained in the first law of thermodynamics. Means what's the first law of thermodynamics states that is conservation of energy, that is energy can neither be created nor be destroyed. It can be changed from one form into another form. And the second law of thermodynamics will explain about entropy. But the concept of enthalpy is followed by the first law of thermodynamics. Okay. So option A is the correct answer for question number 75. So next question, that is 76, based on the Ramachandran plot. So here Ramachandran plot predicts which of the following parameters of a protein. So here anyhow the answer for this question is option D, dihedral angle. So what are dihedral angles? That is psi and also phi. Psi against the phi of amino acid residues. So here this Ramachandran plot is used to predict the secondary structure of proteins, that is based on this parameter dihedral angles psi and also phi. Okay, so this is question number 76. So now move on to the next question, that is 77. Okay, so in nucleotide, sugar and base are linked to each other by, okay, so for example nucleotide which contains ribose sugar. So this ribose sugar is form a bond which contains three phosphate group. For example, adenine, guanine, cytosine, thymine, AMP, ATP, ATP, like this. Okay. So ATP means what? Adenosine, ATP, adenosine triphosphate, which contains adenosine, that is nucleoside. Nucleoside contains ribose sugar, which is linked with the base. So what are the nitrogenous bases? Adenine, guanine, cytosine, as well as thymine. Okay, any one of the base which is linked with the ribose sugar. So here in the question they ask that is sugar and base are linked together by glycosidic bond, that is glycoside bond. So here the with the phosphate and sugar the bond is phosphodiester bond, but here they are asking about the sugar and base linkage. So that's why option B, glycoside bond is the correct answer. Okay. So now move on to the next question, that is question number 78. Which of the following enzyme is not involved in galactose metabolism? Okay. So now write about metabolism of galactose. So galactose is converted into galactose 1 phosphate, and this galactose 1 phosphate converted into UDP galactose, and this UDP galactose is converted into UDP glucose. So enzymes for the conversion of galactose into galactose 1 phosphate, that is galactokinase, and the enzyme which converts galactose 1 phosphate to UDP galactose, that is galactose 1 phosphate uridyl transferase, and conversion of UDP galactose to UDP glucose, that is UDP galactose 4 epimerase. Okay, galactokinase is there. Yes, option D. Galactose 1 phosphate uridyl transferase is there in option A. UDP galactose 4 epimerase is there in option B. But here there is no function of glucokinase, that is addition of phosphate group to glucose, but here the phosphate group is added to galactose, not to glucose. So that's why option C is correct answer, because glucokinase is not involved in galactose metabolism. Okay, so this is about question number 78. So now move on to the next question, that is question number 79. So which of the following forces is most favorable for protein folding? Okay, so here for the protein folding, the major forces which is involved is, that is hydrophobic interaction. So that's why option A is the correct answer for the question number 79. Okay, anyhow van der Waals force hydrogen bond is also involved, but the most favorable interaction is hydrophobic interaction. Okay, so now move on to the next question, that is question number 80. So which of the following is not true about lipid rafts? So usually lipid rafts are small microdomains which is ranging from the size 10 to 200 nanometer in size. Okay, so here lipid rafts are mainly constituted of cholesterol. Yes, lipid rafts are assembled in Golgi complex and directly sent to plasma membrane. Correct. So one lipid raft is large enough to be seen under microscope. No. So here this is very small, which is ranging from 10 to 200 nanometer. So that's why which is very difficult to visualize. So that's why it should be visualized under which one, fluorescent molecular probes. Okay, and also lipid rafts when activated forms into clusters. Correct. Okay, so here option C, that is one lipid raft is large enough to see under the microscope, that is option C is the correct answer, which is not true about lipid rafts. Okay, so now move on to the next question, that is question number 81. So fluidity of the plasma membrane is mostly decided by the following constraint. Option A, phospholipid. Option B, sphingolipid. Option C, cholesterol. Option D, protein. So here fluidity which is decided by the cholesterol, because cholesterol prevents membranes from freezing, which will maintain the membrane fluidity. That's why option C is the correct answer for the question number 81. So now move on to the next question, that is question number 82. The only electron carrier in electron transport chain which is not, you can see here, not a protein bound prosthetic group, that is which does not bind with the protein as a prosthetic group. So cytochrome C, yes, which contains heme as a prosthetic group. So iron sulfur cluster, yes, it has NADH reductase, it also has prosthetic group. But coenzyme Q is a prosthetic group that does not bind to a protein Okay, so that's why coenzyme Q also called as ubiquinone. This is the correct answer for question number 82. So now move to the next question, that is question number 83. That is match the following. So yeah, we have to match this. So microtubules, actin, lamin, keratin. So here correct answer for this match the following. Option A, because we don't have much time to discuss about individual concept. So that's why option A is the correct answer for this question. Microtubules, spindle formation, actin, muscle contraction, and lamin, cell adhesion, and keratin, desmosomes. Option A is the correct answer for this question. So the next question, that is question number 84. Okay, so chromatin condensation in a cell to form fibers requires. Okay, so here deacetylation, which will leads to the chromatin condensation, which will form the fiber. So that's why option B, deacetylation of histone tails, that is by the enzyme histone deacetylase, which will result in more closed chromatin structure, that is condensed chromatin structure. Okay, option B is the correct answer for question number 84. So next, 85th question, that is the following cell cycle transition which is not an irreversible process, that is option B is the correct answer. G1 to G0 phase. Anyhow, G1 S phase is reversible, irreversible. Metaphase, anaphase also irreversible. Anaphase, telophase also irreversible. But this G1 to G0 phase, which is also called as quiescent phase, that is senescence phase, which is not an irreversible process. Option B is the correct answer for question number 85. So the next question is 86. The following statement is not correct about auxotropes. So based related to the auxotropes, which is the not correct statement, they are unable to synthesize all the vital nutrients. The first option itself, it is wrong, means correct for this question, but usually auxotropes are able to synthesize all of the vital proteins. Okay, so next, once again, yeah, option A, they are able to synthesize all the vital nutrients. TRP1 is an auxotropic marker gene, correct. They grow in minimal media, not in supplement media, correct. Auxotropic mutants can be isolated by filtration enrichment technique, correct. So option A is the correct answer for the 86th question, but this statement is not correct about auxotropes. Okay, so the next question, that is question number 87. The amino acid that is encoded by only a single triplet code. So here two amino acids are there which can encoded by only a single triplet code, that is methionine and also tryptophan. So all other contains multiple triplets of the codon, but these two amino acids encoded by only a single triplet code. So here methionine is there in the option, so that's why option D, methionine, is the correct answer. Okay, so now move to the next question, that is question number 88. Okay, 88. Which of the following double strand repair mechanism occurs throughout the cell cycle? So here, when it comes to double stranded repairs mechanism, the non-homologous DNA end joining, that is at any time in the cell cycle which will breaks the double stranded breaks can be repaired, which is repaired by the double stranded mechanism, that is non-homologous end joining or NHEJ. So option D is the correct answer for question number 88. So next one is 89 question, that is histone acetylation increases transcription of gene because that is it loosens the DNA histone complex to make it accessible to RNA polymerase, that is allowing for easier gene expression. It has to loosen the DNA histone complex, which helps to access for the RNA polymerase enzyme, that is for the transcription process. Okay, so option D is the correct answer for question number 89. Okay, so now move on to the next question, that is question number 90. Okay, so The eukaryotic RNA polymerase that transcribes pre-mRNAs is. So here, RNA polymerase which will transcribe three different RNA, that is tRNA, rRNA, mRNA, tRNA. Just remember like RMT, that is RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. So here they have transcribed the pre-mRNA where M is coming under the RNA polymerase 2. So that's why RNA polymerase 2, where RNA polymerase 2 is there, that is option B, RNA polymerase 2 is the correct answer. And RNA polymerase 1 for the rRNA synthesis and RNA polymerase 3 for the tRNA synthesis. So here in this, for this question, option B, RNA polymerase 2 is the correct answer. Okay. So next question, that is question number 91. Which organelle is responsible for the modification, sorting, packaging of proteins for secretion or intracellular use? So when it comes to the packaging, the one and only organelle which is involved, that is Golgi apparatus. So option C is the correct answer for the question number 91. Okay. So now move on to the next question, that is once again, is question number 92. Which kind of the following post-translational modification increases the stability of the proteins? So here the stability of the proteins were increased by N-linked glycosylation, because in the N-linked glycosylation, sugar will attach to asparagine, which will aid in proper folding and protect against the degradation, and this will enhance the stability. Okay, that's why option B is the correct answer for question number 92. So next question, that is question number 93. In the RNAi regulatory pathway, the dicer enzyme cleaves. Okay, so that is RNA interference, RNAi means. So here what happened, the dicer, one of the enzyme. Okay, so this dicer enzyme will, the function of this dicer enzyme, that is double strand, which will cleaves the double stranded RNA into short strands. Anyhow, enzyme, the function of the enzyme is what, which will involved in the cleaving process. Okay, so here this dicer enzyme which is present in RNAi, okay, so which will break down or cleaves the double stranded RNA into short strands. Okay, option C is the correct answer. So then when it is cleaves into short strands, which then guide into RISC complex, that is RNA inducing complex. Okay, so next question, that is silencing complex. So the next question is question number 94. So bacterial pathogen enter the host through their appendages, which are, okay, so here option B is the correct answer. That is FIMH gene product that binds with the trimannose residues. That is what takes place when the bacterial pathogen enters the host. It has to attach to the cell, that is host cell. So for the attachment and the appendages, this FIMH gene product will binds with the trimannose residues, helps for the pathogenesis. Okay. So now move on to the next question, that is question number 95. Sorry. Okay. So 95 question. The following GPCR, that is G protein coupled receptor signaling, does not lead to opening of ion channel. So in which, in these GPCR signaling pathways are there. So here, which does not lead to opening of ion channel. So okay. So option A, that is muscarinic acetylcholine receptor. So this is also opening the ion channel by GPCR signaling. Then epinephrine receptor, this one also opens the ion channel, and rhodopsin also leads to opening of ion channel. So option D, that is nicotinic acetylcholine receptor, which is ligand gated ion channels, means they directly allow the passage of ions when bound by acetylcholine or nicotine. So that's why option D is the correct answer for the question number 95. Okay. So now move on to the next question, that is question number 96. Okay, match the following. So here in the match the following, based on, that is TFG, beta cytokine receptor, RAF, RAS. So for the 96 question, option B is the correct answer. That is um, for A, that is TFG beta, SMAD. For B, that is a cytokine receptor, JAK, that is JAK-STAT pathway. And option C, that is for RAF, serine threonine kinase. And that is RASMAP pathway, that is D, that is uh, tyrosine kinase, which is a receptor. Okay, so this is about the match the following in the 96 question. Just go through the options, okay, because we don't have that much time to explain each thing. Okay, so now move on to the next question, that is question number 97. Here, this one 97 question is also match the following. That is about cell junctions, adherents, desmosomes, tight junctions, hemidesmosomes. Okay, so answer for this question number 97 is option D, that is adherent junction, CAM, and desmosomes, placoglobin, and tight junctions, Claudin 1, and hemidesmosomes, that is integrins. Option D is the correct answer for question number 97. So now move on to the next question, that is question number 98. Here, this one also, match the following. Okay, some tumor suppressor genes are there: retinoblastoma gene, C junction, P53, TFG beta. Okay, so here the correct answer for this question number 98 is option D. That is retinoblastoma gene, transcription factor, C junction is progress through cell cycle, P53 that is signal transducer, then TFG beta, that is cell cycle checkpoint. Okay, so anyhow, even though somehow, for example, P53 is also related to this one, cell cycle checkpoint, because which is involved in the cell cycle of cancer. Sometimes all the options are synced with all the questions are synced with all the options. So we have to wait till final key answers will release. But this is how to tell. Somehow these options are correct. Okay, just guessing. That's it. So next one, that is 99, question number 99. Let's move on to that. Okay, 99 and 100. Last two questions. We are at the end. So the following statement about antibody engineering is not correct. Okay, so this involves monoclonal antibodies, correct. This involves modification of structure of antibody, correct. Involves modification of sequence of antibody, correct. This involves polyclonal antibodies only, not polyclonal antibodies only, because it involves monoclonal antibody also. So that's why option A is the correct answer, which is not correct about antibody engineering for the question number 99. So the last question, which is related to COVID-19, that is COVID vaccine. Covaxin is a vaccine which is option A developed by India, administered against SARS virus, no. NAU and mRNA vaccine, no. Advised for persons with anaphylaxis, no. So here option A is the correct answer, which is developed by India. So here this Covaxin vaccine is there, right? This Covaxin is a whole inactivated vaccine, whole inactivated virus vaccine, which is based on the COVID-19 vaccine developed by a Bharat Biotech, that is developed by India, which is collaborated with the ICMR. That's why option A is the correct answer for question number 100. So this is about KSET LifeScience 2024 solution video. If I made mistakes in any question, let me know in the comment box. Okay, I'll correct it. And if any doubts related to this KSET LifeScience question paper, not only KSET, if you people want me to solve general paper also, that also let me know in the comment box. So thank you so much for watching, and keep watching, keep supporting me. Subscribe my channel to more videos like this. And that's it for today's video. So thank you so much.